The Shroud of Turin. To believers, it bears the miraculous image of Christ crucified. But tests done in 1988 showed it wasn't that old.
The Shroud of Turin. That's the linen cloth believed to bear Jesus' imprint as he was being prepared for burial. And now there's new research.
Hidden behind bulletproof glass in Turin Cathedral lies a linen cloth that should be nothing more than fragile history. And yet, it is far more than it seems. Imprinted on its fibers is the image of a crucified man, an image no artist, no chemical, no known process can fully explain. For centuries, the world has argued over what it is. Now, DNA embedded deep within the cloth is telling a story that no one expected, one that could rewrite everything we thought we knew. The Turin Shroud mystery.
The controversial and mysterious Shroud of Turin, revered by millions, hasn't been seen by the public for 10 years.
Let me take you back for a moment, because you cannot understand what was found inside the shroud unless you first understand the century of war that has been fought over its surface. The Turin Shroud first appeared in the historical record in the 1350s when a French knight named Geoffroy de Charny displayed it in a small church in Lille, France. Almost immediately, it was controversial. A local bishop wrote to the Pope claiming it was a forgery, painted by a clever artist. The church never officially endorsed it as authentic, but the faithful did not need official endorsement. The image was too haunting, too detailed, too unlike anything anyone had ever seen. It showed a bearded man with wounds consistent with Roman crucifixion, nail marks through the wrists, not the palms, which is anatomically correct for crucifixion, but was not widely known in the medieval period. Scourge marks across the back, consistent with a Roman flagrum. A whip tipped with lead weights and bone fragments. A wound in the side consistent with a Roman lance thrust. Blood flows that follow the exact paths gravity would dictate for a body in a crucified position. Whoever or whatever created this image understood Roman execution with a precision that medieval Europe simply did not possess. In 1898, an amateur photographer named Secondo Pia was given permission to take the first photograph of the shroud. And what happened next in his darkroom changed the trajectory of this story forever. When he developed the photographic negative, he nearly dropped the glass plate. The image on the shroud reversed into a perfect positive.
Jesus or not really is a scientific question. It's just since the advent of photography, when the shroud photographed the first time,
the faint, ghostly, barely visible figure on the cloth became a detailed, three-dimensional-looking portrait of a bearded man with closed eyes and an expression of profound stillness. A photographic negative, created centuries before the invention of photography. But think about what that means for a moment. Whoever or whatever made this image encoded information into the cloth in a format that could not be read by the human eye, that could not be decoded by any technology available until the 19th century. And that, when finally revealed, produced a portrait of startling realism. That single discovery transformed the shroud from a devotional curiosity into a scientific obsession, because no medieval forger could have created a negative image. The concept did not exist. The technology to verify it did not exist. It would be like finding a QR code carved into a Roman column. The information format belongs to a different era entirely. Then came 1988, and what many believed would be the final word. 3 independent radiocarbon dating laboratories in Oxford, Zurich, and Arizona tested small samples cut from one corner of the shroud. Their conclusion was unanimous and delivered with the confidence of institutions that do not expect to be questioned. The linen dated to between 1260 and 1390 CE medieval. End of discussion. Case closed. The shroud was a fake. Headlines around the world declared it in bold type. Textbooks updated accordingly. Museums revised their displays. The scientific establishment congratulated itself on having settled one of history's most persistent questions with the clean. a dispassionate authority of radiometric chemistry. The debate, everyone assumed, was over, except it was not over, not even close. Because almost immediately, other scientists, many of them with no religious stake in the outcome whatsoever, began pointing out serious problems with the 1988 test. The samples had been taken from a single corner of the cloth, a corner known to have been handled more than any other part, a corner that had been repaired and rewoven after the devastating fire of 1532. Where textile analysts demonstrated that the sampled area contained cotton fibers interwoven with the original linen, clear physical evidence of a medieval reweaving patch that would have contaminated the dating results with younger material. A peer-reviewed statistical analysis published in 2005 by Raymond Rogers, a chemist from Los Alamos National Laboratory, showed that the radiocarbon dates across the 3 laboratory samples were not homogeneous, and they did not come from a single uniform piece of fabric. The corner they tested was chemically different from the rest of the shroud. Its vanillin content, a compound that degrades predictably over time, was significantly higher than the main body of the cloth, suggesting it was indeed younger. In other words, they may have dated the repair, not the cloth. The 1988 result, the one that was supposed to end the debate forever, may have been measuring the wrong thing entirely. And the institutions that declared the case closed have never agreed to retest. And then there was the image itself. Despite decades of effort, no one has been able to reproduce it. Not with paint, not with scorching, not with acid, not with any photographic technique. The coloration exists only on the topmost fibers of the linen, penetrating to a depth of approximately 200 nanometers, thinner than a single bacterium. It has no directionality, no brush strokes, no pigment particles. It behaves like a superficial dehydration of the cellulose, as if something flash-heated the outermost surface of the cloth without affecting anything beneath it. In 2011, Italian researchers at ENEA, the National Agency for New Technologies, attempted to replicate this effect using an excimer laser and concluded that reproducing the shroud image would require a burst of vacuum ultraviolet radiation with a total power output of several billion watts. Billion, with a B. And no known medieval or ancient technology could produce that. Frankly, no modern technology can produce it either. Not at that scale and that precision. That is the world the DNA researchers walked into. A relic that carbon dating says is medieval. That image analysis says is impossible to forge. That anatomical detail says understands Roman crucifixion better than any medieval scholar, and that nobody, in over a century of trying, has been able to explain. Discovery of the hidden DNA. In 2015, and something happened that should have made front-page news around the world, but somehow barely registered outside of academic journals. A team of researchers led by geneticist Gianni Barcaccia at the University of Padua in Italy published a study that, if you understand what it actually found, quietly detonates beneath everything we thought was settled about the Shroud of Turin. Here is what they did. During a major examination of the shroud in 1978, a team of American scientists known as STIRP, as the Shroud of Turin Research Project, was granted direct access to the cloth for 5 continuous days. 120 hours of testing. They photographed it, they sampled it, they pressed adhesive tape against its surface to collect fiber samples, and they vacuumed it, carefully collecting dust and particulate matter from between the fibers using specialized equipment designed to capture microscopic material without damaging the linen. Those vacuum samples were preserved and stored. For decades, they sat in controlled environments, waiting for technology to catch up with ambition. In 2015, it finally did. Barcaccia's team applied next-generation genomic sequencing to these deep fiber samples. Now, let me be very precise about what that means, because this is where most people get confused and where most critics attack. Finding DNA on the surface of the shroud would mean absolutely nothing. Any cloth that has been handled by priests, displayed to crowds, kissed by pilgrims, unfolded and unfolded by conservators, and stored in churches across Europe for 7 centuries, is going to be covered in genetic material from every person who ever breathed near it. That surface contamination is noise. It is meaningless. Everyone agrees on that. What Barcaccia was looking for was something entirely different. DNA that was not sitting on the surface but embedded within the fiber structure itself. Genetic material trapped inside the microscopic gaps between cellulose chains in the linen, material that was incorporated at or near the time of the cloth's creation, locked in place by the same processes that preserve ancient DNA in bone and tooth enamel. Material that no casual contact could deposit and no ordinary cleaning could remove. What they found stunned even the researchers themselves. The human DNA told a story that no medieval forger could have written. Mitochondrial sequences belonging to haplogroups from across the known world. European lineages, yes, exactly as you would expect from centuries of handling in France and Italy, but also lineages characteristic of the Middle East, of North Africa, of East Africa, and most unexpectedly, of East Asia. The shroud's genetic fingerprint did not read like a cloth that had spent its entire life in medieval Europe. It read like a cloth that had traveled extensively through the ancient Near East, accumulating biological traces from populations along the way before arriving in the West. But it was the plant DNA that truly changed the conversation. Barcaccia's team identified genetic sequences from plant species native to the Mediterranean, from Central Europe, and critically, from the Middle East. Several species matched the flora of ancient Jerusalem and the surrounding Judean hills. Others corresponded to plants found along the historical trade and pilgrimage routes connecting the Levant with Edessa in southeastern Turkey, then onward to Constantinople, and finally to Western Europe. The botanical evidence mapped a journey, and that journey matched, with remarkable precision, the exact route that historical documents describe for the shroud's movement from Jerusalem through the centuries. Edessa, as where a mysterious cloth bearing a face was documented as early as the 6th century. Constantinople, where a burial cloth matching the shroud's description was recorded in the imperial relic collection before the Fourth Crusade in 1204. And then France, where it appeared in the possession of Geoffroy de Charny in the 1350s. If the shroud were a simple 14th century forgery manufactured in medieval France, its embedded DNA should reflect 14th century France, local plants, local human populations, a clean, geographically limited genetic signature. Instead, the genetic profile tells a story that spans 2,000 years and thousands of miles, a story that the forger would have had no motive to create, no method to fabricate, and no knowledge to even conceive of, because the science of genomics would not exist for another 600 years. The DNA is not proof of authenticity, but it is proof that the simple explanations have stopped working. And when the simple explanations fail, the complicated questions begin. Shroud DNA evidence. Here is where things get genuinely strange, and I need you to stay with me, because the next part of this story requires holding 2 ideas in your head simultaneously, one scientific and one almost philosophical, and letting them collide. Among the human DNA sequences recovered from the deep fiber samples, the researchers identified mitochondrial haplogroups that are characteristic of populations from the Druze communities of the Middle East, from ancient Sephardic Jewish lineages, and from populations in the region around the Dead Sea. These are not the genetic signatures of medieval European handlers. These are genetic fingerprints consistent with a 1st century Judean population, the kind of DNA you would expect to find on a burial cloth used in Jerusalem 2,000 years ago, and the kind of DNA that a medieval forger in France would have had absolutely no way to acquire.
DNA testing are in. This unprecedented analysis has uncovered some astonishing information. The genetic material tested from the shroud is mitochondrial DNA.
No reason to plant, and no technology to embed within the fiber structure of a linen weave. But what caught the attention of geneticists was not just where the DNA came from, it was how it was preserved. Normally, DNA degrades rapidly in textile fibers. Exposure to light, moisture, and microbial activity breaks down nucleotide chains within decades, certainly within centuries. Finding readable genetic sequences in a cloth that is either 700 or 2,000 years old, depending on which dating you accept, is already unusual. Finding sequences this diverse, this well-preserved, embedded this deeply, suggests that something about the conditions surrounding this cloth at the time of the DNA's deposition was extraordinary. Either the cloth was sealed in an environment with virtually no biological activity, no light exposure, no moisture exchange for an extended period immediately after contact, or something about the event that created the image also created conditions favorable to DNA preservation in ways we do not yet understand. Some researchers have gone further, pointing out that the pattern of genetic diversity on the shroud does not resemble random contamination at all. Random contamination would produce a noisy, disorganized genetic profile with no geographic coherence, a statistical soup of whatever happened to land on the surface. What the shroud shows is structured diversity, genetic signatures that cluster geographically and historically in ways that tell a coherent story. A story of origin in the ancient Near East, of storage and transport through specific historical corridors, and of arrival in medieval Europe. The DNA does not read like an accident. It reads like an itinerary. And it is as if the cloth recorded its own biography in a genetic language that we could not read until the 21st century. And now that we can finally read it, the biography matches the legend with a precision that makes coincidence an increasingly uncomfortable explanation. The theological implications are obvious, and I am going to be honest with you about them rather than pretending they do not exist. If the DNA embedded in the shroud's fibers genuinely belongs to a first-century Judean male, and if the image on the cloth was created by an event energetic enough to produce a burst of radiation measurable in billions of watts, then what we are looking at is physical evidence of something that science has no current framework to explain, and that one particular religious tradition has been claiming for 2,000 years. I am not telling you what to believe. I am telling you what the data says. And the data is uncomfortable for everyone, believers and skeptics alike, because it refuses to fit neatly into either narrative. Implications for history and faith. The reaction to Barcaccia's findings was exactly what you would expect from a discovery that threatens to destabilize a century of settled opinion. Some scientists praised the methodology and called for expanded testing with next-generation sequencing tools. Others dismissed the results as contamination artifacts, arguing that no amount of genomic analysis could overcome the fundamental problem of a relic handled by thousands of people over hundreds of years. The Vatican said very little publicly, which is what the Vatican tends to do when science gets close to something it considers sacred. They did not endorse the study. They did not condemn it. They simply let the silence do its work, which, if you have followed Vatican communication strategy over the centuries, you will recognize as a very deliberate choice. And the popular media mostly ignored it, because a nuanced genetic study with carefully hedged conclusions is harder to sell as a headline than a definitive declaration of authenticity or fraud. The world wanted a yes or a no. The DNA gave them a maybe that leaned in a direction nobody was comfortable with. But the implications for those willing to sit with them are profound. Consider what the DNA evidence means for the carbon dating controversy. If the embedded genetic material maps a journey from first-century Jerusalem through the ancient Near East to medieval Europe, then the cloth itself must have existed long before the 1350s. The carbon dating result, which placed the shroud's origin in the medieval period, becomes increasingly difficult to reconcile with a genetic profile that tells a story spanning 2 millennia. Either the DNA is wrong, or the carbon dating sampled the wrong part of the cloth. One of these two scientific conclusions must give way. And at the moment, the carbon dating has far more methodological problems than the genetic analysis does. For historians, the DNA evidence offers something unexpected.
Those are the samples from the Turin Shroud.
Wow.
Here, clots collected from the shroud.
Clots, meaning blood.
Blood-like particle.
A potential tool for tracking the physical movement of relics through the ancient world. If the shroud's genetic profile can be matched to specific geographic regions and historical periods, then it becomes possible, at least in theory, to verify or refute the traditional accounts of its journey. Did it really pass through Edessa, as the historical record suggests? The DNA should contain traces of species and populations characteristic of that region. Was it really housed in Constantinople before the Fourth Crusade? The genetic evidence should reflect that. For the first time, we have a method that could transform the history of relics from a matter of faith and tradition into a matter of testable evidence. For the faithful, the discovery presents a paradox that is both thrilling and terrifying. If science confirms that the shroud wrapped a first-century Judean man whose death left an image that required billions of watts of ultraviolet radiation to create, that is not a comfortable confirmation of faith. It is an intrusion of the measurable into the domain of the miraculous. It takes something that was supposed to be believed and makes it something that can be analyzed. And analysis, by its very nature, it is never finished. What science gives, science can also take away. The DNA that seems to support authenticity today could, with further testing, reveal something that complicates the picture tomorrow. Faith, by definition, does not require evidence. When evidence arrives, it does not strengthen faith. It replaces faith with something else entirely. And not everyone welcomes that trade. The future of shroud research. Here is what we know is coming. Because the technology is already here, and the pressure to use it is only growing. Next-generation genomic sequencing is orders of magnitude more sensitive than what was available to Barkaccia's team in 2015. Techniques like nanopore sequencing can read individual DNA molecules in real time, extracting usable genetic data from samples so small they would have been invisible to earlier instruments. Ancient DNA analysis. The same discipline that sequenced the Neanderthal genome from 40,000-year-old bone fragments has matured to the point where extracting a complete genetic profile from a 2,000-year-old linen cloth is no longer speculative. It is achievable. The question is not whether the technology exists. The question is whether anyone will be granted access to use it. In 2002, the shroud underwent a major conservation effort during which the backing cloth was removed and the surface was cleaned. Material collected during that process is believed to be held in multiple locations, not all of which are under Vatican control. Independent researchers have been requesting access for years, and the political landscape around shroud research is shifting. A new generation of scientists, less burdened by the partisan loyalties of the 1988 carbon dating war, is approaching the relic with better tools and fewer preconceptions. Several research proposals are currently circulating that would combine genomic analysis with advanced imaging techniques, including synchrotron radiation scanning and X-ray fluorescence mapping, to build a comprehensive molecular portrait of the shroud that goes far beyond what any previous study has attempted. There is also growing interest in the intersection of genetics and archaeology more broadly. The success of ancient DNA studies at sites like Pompeii, where researchers extracted complete human genomes from victims preserved in volcanic ash, and at Denisova Cave in Siberia, where a single finger bone yielded an entire unknown human species, has demonstrated that genetic material can survive extreme conditions when the circumstances of preservation. DNA has been recovered from permafrost mammoth tusks, from Egyptian mummification resin, from the dental pulp of medieval plague victims. If the event that created the shroud image also created conditions favorable to DNA preservation, as some researchers suspect, then the shroud may contain the most well-preserved ancient genetic material outside of permafrost or volcanic deposits. That would make it not just a religious artifact, but a scientific treasure of the highest order. A molecular time capsule containing biological information from 1st century Jerusalem that exists nowhere else on Earth. The tension between science and faith is real, and it is not going away. But perhaps the most honest way to hold that tension is to acknowledge that they are not actually asking the same question. Science asks what happened. Faith asks what it means. The DNA in the shroud can tell us where this cloth has been, who touched it, what plants surrounded it, and possibly what biological event created the image on its surface. It cannot tell us whether that event was divine. That question belongs to a different kind of investigation, one that no microscope will ever resolve. And maybe that is exactly as it should be. And so the Shroud of Turin remains, defying explanation, resisting certainty, holding secrets woven into fibers older than most civilizations. Science can measure, history can speculate, and faith can wonder. But none can fully unravel what it remembers. What messages are hidden in its threads, waiting for eyes and minds ready to read them? And when we finally understand, will it answer the questions we ask? Or ask new ones we are not yet ready to hear?